Hello and welcome to this NASCalf tutorial. In this video we'll look at how NASCalf models protein and amino acid nutrition in young calves. Protein nutrition in calves is still not fully understood, particularly the flow of metabolizable protein and amino acids from dietary and microbial sources. Early in life, the calf functions essentially as a monogastric, with most amino acids coming from milk that bypasses the rumen and is digested in the abomasum. As the rumen develops, this shifts, and an increasing proportion of amino acids reaching the small intestine are derived from microbial protein, primarily bacteria, and eventually protozoa once a stable rumen environment is established. NASCalf models this transition using assumptions about changing contributions of microbial protein and rumen undegradable protein based on the 2021 NASM publication. However, the 2021 NASM does not fully adjust changing flow of RUP and microbial protein. NASCalf assumes the ratio of RUP to RDP changes with changing rumen development until a mature rumen environment is achieved. The program also assumes that all RDP is converted to microbial protein, which is likely a simplification of the actual events occurring in the calf's rumen. The flow of microbial protein is assumed to be all bacterial until forage is fed, which helps stabilize the rumen pH and allows protozoa to begin to contribute to microbial amino acid supply. All of these calculations are outlined in the Calf Notes technical seminar entitled Reimagining Protein Nutrition in Young Calves. You can find this presentation at Calf Notes Academy at academy.calfnotes.com. Amino acid requirements in the model are calculated using a factorial approach, accounting for both maintenance and growth. These calculations follow the now 2021 NASM guidelines and are based on empty body weight gain and the composition of that gain. To help evaluate these dynamics, NASCalf provides two diagnostic tools. The first is the protein report, which summarizes maintenance requirements such as endogenous urinary losses and metabolic fecal nitrogen, and estimates amino acid needs. It also shows metabolizable protein supply from rumen undegraded protein and microbial sources, separated by liquids, grains, and forages. The second tool is the nutrient supply window, which compares amino acid supply to requirements. This includes contributions from liquids, dry feeds, and rumen undegradable protein, and microbial protein, along with their respective amino acid profiles. The model then calculates total supply and compares it to requirements for maintenance and growth, reporting any deficits or excesses. It's important to recognize that these calculations rely on a number of assumptions due to limited published data. As a result, they should be viewed as estimates only that are directionally useful rather than precise predictors of amino acid limitations. However, they provide a valuable framework for understanding protein nutrition during the calf's transition from a monogastric to a functional ruminant. Well, thanks for watching, and be sure to explore other NASCalf tutorials to learn how to get the most out of the program.